Everyone, and welcome to the Global Immunotalk. My name is Matteo Iannacone. I'm one of the co-organizers. And before we start, let me remind everyone that next week we're going to have Veit Hornung as our guest. So today is my distinct pleasure to introduce Michelle Linterman, who is a group leader at the Braham Institute and a fellow of Churchill College in Cambridge, UK. Michelle is originally from Waikanea in New Zealand, and she obtained her PhD at the Australian National University, where she trained with Carola Vinuesa. In Carola's lab, Michelle described the role for follicular helper T cells in autoimmune disease and characterized key cytokine and co-stimulatory requirements for this. She then did undergo postdoctoral training with Ken Smith at the University of Cambridge, where she continued to dissect the cellular and molecular mechanisms important for follicular helper T cell biology in both mice and in humans. And she also discovered that suppressive follicular regulatory T cells act within germinal centers. In her lab, Michelle focuses on how different cell types collaborate in the germinal center to generate a robust antibody response following vaccinations. And for that, she cleverly combines advanced mouse models with human vaccination studies. And for her work, Michelle has received numerous prizes and awards, including grants from the European Research Council. She's a Lister Institute Prize Fellow, as well as an EMBO Young Investigator. And so welcome, Michelle, and I'm very much looking forward to your talk, which is entitled Aging and the Response to Vaccination. But before we start, Michelle, you know, it is a tradition of the Global Immunotalk to ask a question to get to know you a little bit better. And the question we have chosen for you is, which scientist inspired you the most?
Thanks, Matteo. I am extremely lucky in that I work with and have worked with many inspiring scientists, but the person who's had the most impact, who's the most inspiring, is my PhD mentor, Carola Vinuesa. So not only is she incredibly intelligent, dedicated, passionate and hardworking, she's also an amazing person, and I can only aspire to be like her.
I couldn't agree more. Thanks, Michelle, for sharing, and the floor is yours.
Thank you, and thank you for having me. Thanks everybody for coming. Today, I'm going to focus on aging, and our research on aging is not done by me, but it's done by an amazing team of scientists. These are members of the team, past and present, and so I just want to say up front that good science only comes from the hard work of an amazing and dedicated team, and it's the best part of my job to work with these talented scientists. Now, aging is something that if you're young, you may not be that interested in, but you will become more interested in aging as you get older. But it's an important problem, and it's important because as we get older, we're more likely to have poor health outcomes after infection. And this is demonstrated by the in-hospital mortality rate for two respiratory infections, COVID-19 shown in blue and influenza virus shown in red. And as you can see, that with each decade of life, the in-hospital mortality rate increases. Now, we're really lucky in that we have an established intervention for preventing infectious disease, and that's vaccination. And vaccination has been incredibly successful. It is the reason that we have emerged from the global pandemic, and the reason that many of us survive childhood, because the immunization program for children is incredibly effective. However, as we get older, the protection provided by vaccination decreases, and this is really exemplified in these pie charts that show the protection provided by a single dose of the seasonal influenza vaccine. So in younger adults, the protection against infection is about 80%, and this drops to less than a quarter in individuals over 70 years of age. Now, the reason that this occurs is likely because older bodies are less capable of forming antibodies against vaccine components. So this is some of our data that clearly show that the titer of antibodies against the seasonal influenza vaccine is lower in older individuals. And this is also true for the next generation of vaccines, including the COVID-19 mRNA vaccine. And I show these data, which are not mine. They're taken from a preprint from a New Zealand cohort, Vaccine Alliance Aotearoa New Zealand, that clearly demonstrate that with age, antibody titers after 2 doses of the Pfizer mRNA vaccine, antibody titers decrease. This is also true in the Oxford AstraZeneca vaccines. This is an adenoviral vectored vaccine. And you can also see that the titers of receptor binding domain antibodies decline with age. And what's really interesting for me is that this is clearly a phenomenon that's been well described in multiple human cohorts for multiple vaccines. And this can also be modeled in aged mice. So what do I mean by an old mouse? So for us, this is between 22 and 24 months of age, so nearly 2 years old. And at the onset of the global pandemic, we collaborated with Theresa Lam at the Oxford Jenner Institute to run a preclinical study in our aged mice to determine how well their vaccine worked. And what was really nice is because these preclinical studies were run just in advance of the human trials, we could compare the response in aged mice and in older people and show that aged animals do phenocopy this reduced antibody titer after vaccination that's seen in older people, making them a useful preclinical model for aging and also to understand how the immune system changes with age. And what we really wanted to know was what causes the poor antibody response in older bodies? What really underpins this impaired capacity to mount an antibody response? Now, within secondary lymphoid tissues after vaccination, there are 2 pathways to generating antibody-mediated immunity. Both start with B cells being activated by antigen. They then migrate to the border of the T cell area and the B cell area in secondary lymphoid tissues, where they meet T cells that have been, CD4 T cells that have been primed by professional antigen presenting cells, so-called Tfh precursor cells. The T cells and B cells interact at the TB border, and they exchange signals in a bidirectional manner, and then the B cell gets to make a cell fate choice. So it undergoes a few rounds of proliferation, class switch recombination, and then either differentiates into short-lived antibody-secreting plasma cells, memory B cells, or can migrate into the B cell follicle to seed the germinal center response. Within the germinal center, B cells undergo intense proliferation and mutation of the genes that encode their B cell receptor. Now, this mutational process, whilst targeted to the B cell receptor genes, is random. So you need a process of selection to ensure that the B cells that are, that these mutated B cells are fit enough to provide a good protective antibody response. And to do this, they are tested on 2 different types of cells. First, they move over a network of follicular dendritic cells. Now, these are mesenchyme-derived stromal cells that form the light zone of the germinal center, and they present immune complexes on their surface. And these cells are able to move over this network of follicular dendritic cells and use their mutated B cell receptors to bind antigen And so if they are able to collect antigen from follicular dendritic cells, they take it up, internalize it, and process it on MHC class, process and present it on MHC class 2 to a specialized population of CD4 positive helper T cells, follicular helper T cells. And the B cells outnumber TFH cells in the germinal center and have to compete with each other for TFH cell help. Those B cells that are able to obtain help from TFH cells can either go back from the light zone to the dark zone in the germinal center, where they undergo further rounds of proliferation and mutation, or they can exit the germinal center as long-lived antibody-secreting plasma cells that home to the bone marrow or memory B cells. And the germinal center response is negatively regulated by a population of follicular regulatory T cells that express FOXP3. So what we wanted to understand in aging is whether this poor antibody production was due to defects in this short-lived antibody-secreting plasma cell pathway or in the germinal center. So these are data taken from our preclinical study of the Oxford-AstraZeneca vaccine. And what was very clear 9 days after immunization is that there was no defect in this formation of early antibody-secreting plasma cells between young and aged mice. They were absolutely identical. The only defect that we did see was that older mice had a slightly higher proportion of IgM positive plasma cells, although still a very small proportion of the total plasma cell pool. What we did note, both by confocal imaging, so these are spleen sections taken 9 days after immunization, and what you can see on the top, outlined by the dotted line, is the germinal center. The B cell follicle is in green that surrounds the germinal center. The follicular dendritic cell network is in white, and proliferating B cells are seen with Ki67. Now you can clearly see that the structure is smaller in aged mice and that the structure looks a little odd. When we enumerated germinal center B cells by flow cytometry in our aged mice, we saw that they were reduced by about half in aging. So what we think is that the defective antibody response upon vaccination is really due to a poor germinal center response rather than that early antibody secreting cell pathway. So what about in people? Can we see an effect or can we see whether the germinal center response is impaired in older people? And one of the challenging things about examining the germinal center response to vaccines in humans is the fact that they take place in secondary lymphoid tissues. So it's something that's very hard to access routinely unless you're doing fine needle aspirates. So what we did for this study was we took advantage of a lot of work really pioneered by a number of different labs that have shown that there's a population of cells, CD4 positive T cells in the peripheral blood that act as a circulating biomarker of follicular helper T cells. And we wanted to track these cells in the blood after vaccination. But what we wanted to do was be able to track influenza-specific circulating TFH cells. And to do this, we recruited a cohort of volunteers, either younger or older adults, who we knew their HLA type and could use class 2 tetramers to track antigen-specific cells, antigen-specific CD4 T cells. And so this is a flow cytometry plot shown here. What we noted was that in all of our volunteers prior to vaccination, we saw a small number of antigen-specific CD4 T cells, and this was consistent with pre-existing antibody to this this H1N1 HA. What you can nicely see is that these cells undergo an expansion in number 7 days after immunization, and this was something we saw in both our discovery cohort, so this was run in the 2014-15 influenza season, and then in our replication cohort that was run in the 2016-17 influenza season. And all of this work was led by Danika, who's a former postdoc in the lab. What we could nicely see is that 7 days after vaccination, there was an expansion of cells that had a circulating follicular helper phenotype, so CXCR5 positive, PD1 positive. And even more interestingly for us, what was obvious both in the discovery and in the replication cohort was that the frequency of circulating follicular helper T cells was reduced in older individuals, really suggesting that the germinal center response is impaired in people. Now, I'd like to show some data that doesn't come from my lab, but was provided by Michaela Lockie, and I'd just like to acknowledge her for sharing her data. She's done a nice series of experiments which were published in Cell this year, where she took fine needle aspirates of lymph nodes from humans 8 days after their third mRNA COVID vaccine. And after she shared the data with me, you could see by reanalyzing these that there's a clear trend to having fewer spike binding, RBD binding germinal center B cells in the lymph nodes of their older volunteers. And this was also observed in the lymph nodes of their volunteers. If you look at the spike binding and RBD non-binding germinal center B cells. So what this really shows is that after vaccination in humans, it really does look like the germinal center response is impaired, as it is in mice. So these are the data from one of our mouse studies done by Alyssa Silva-Cayetano. And in the mouse, what is much easier to do is a nice kinetic study to really ask what are the problems with both the initiation and the maintenance of the germinal center response. And what we noted were two things. So this is the percentage of germinal center B cells on the left and the number on the right. And what's very clear is that there is both a delay in the germinal center response, so it's slower to get started, but also the peak never reaches the same height in aged mice, shown by the open symbols compared to young mice in the black symbols. And so we've done a lot of work to try and understand what contributes to both the delay in the germinal center formation in aging, but also this impaired peak of the response. And that's what I will present today. So the first part of the talk is a published story that really focuses on this delayed germinal center formation. And then the second part is some unpublished work that focuses on what causes the diminished size of the germinal center response in aging. So so one of the first experiments that we did was a heterochronic parabiosis study to really try and understand where the primary defect is. So is the defect in the cells of the immune system that can recirculate, or is the defect a more microenvironmental defect? And so what we did was we surgically joined younger adult mice, aged mice, or we did a heterochronic setup where we surgically conjoined and aged to an younger adult mouse. And the nice thing about this is that the circulatory system of the parabionts joins, and so cells from the aged animal can flow into the young animal and vice versa. But what remains intact in the mice is the resident cells. So for example, the stromal cells, any tissue resident immune cells would stay in the same place. So that allows you to disentangle the age of the circulating cells from the resident cells. And really what we wanted to understand was, is it the age of the immune cells or that of the lymph node that impairs germinal center formation and aging? And so in these experiments, we were looking early after immunization, so 7 days after immunization. What we did was we took the parabios mice and we immunized them on the outer flank, so the non-surgically conjoined side, and measured the germinal center response 7 days after immunization. So I'll just take you through these data. So if you take the mice that are paired with a mouse of the same age, what you observe is that the adult mice, the younger adult mice, have more germinal center B cells than the aged mice. So this is consistent with the previous studies that I've shown you. If you then look in the heterochronic parabiosis, so here they're split into the mouse, the adult mouse on one side and the aged mouse on the other. And what you can clearly see is that in the aged microenvironment, there are fewer germinal center B cells. And this really suggests that it's the age of the lymph node rather than the age of the circulating immune cells that makes that causes the delay in germinal center formation. You can also take these data and break them down further, because we'd use congenic markers to distinguish CD45, we'd use congenic markers to distinguish where the cells came from, whether they were from the younger adult animal or the aged animal. And I'll just walk you through this again. So on the top is the age of the host. So this is the age of the microenvironment. And on the bottom is the age of the B cells. So what you can see is that when B cells come from a younger adult mouse, but they're in an aged microenvironment, they're less likely to become germinal center B cells. And if you look in the other way around, if you look at the capacity of aged B cells to participate in a germinal center response in a young microenvironment, you can see that they do this very well. And so this experiment tells you two things. It first tells you that it's probably the age of the microenvironment that causes poor germinal center initiation in aging. But it also tells you that germinal center B cells, or the ability to become a germinal center B cell, isn't compromised by the age of the B cell. So then this leads to the question of which are the cell types in the lymph node microenvironment that are responsible. And we focused on lymphoid stromal cells. This is a particular area of expertise of Alice Denton, who's a former postdoctoral fellow in the lab who now runs her own lab in London. So the lymph node stromal cells, particularly with respect to germinal center biology, are somewhat neglected compared to TMB cells. But the mesenchyme-derived stromal cells really provide the stage upon which immune cells act. And this is a cartoon from Alyssa Silva-Cayetano in the lab, where she really tries to bring together the stromal and the hematopoietic compartments. And what I'd like to draw your attention to for this talk is two types of stromal cells that are important in the germinal center response. First are the marginal reticular cells that line the subcapsular sinus. So these are stromal cells that will be some of the first to sense antigen as it enters the lymph node. And follicular dendritic cells, which define the light zone of the germinal center, and that cell type that I had said presents antigen to germinal center B cells to allow them to test their mutated B cell receptors. So going back to our parabiosis experiments, we wanted to understand if either of these stromal cell types had impairments in our parabiosis experiments. The nice thing about this is that, again, you can look at the stromal cells which don't move in both adult and aged mice. So these are the mice that are paired with a mouse of the same age. And what you can see is that there are fewer MRCs in the aged pairs. If you then look at the heterochronic parabionts, so the mice that were paired with an animal of a different age, what you can see is that in the aged microenvironment, there's clearly fewer MRCs compared to the young animal. So it looks like there's something going on with the aged MRCs that can't be rescued by circulating cells from a young animal. If you then look at follicular dendritic cells, you again see that there are fewer in aged mice. And then the result for the heterochronic parabionts was quite interesting, because what we saw was that in an aged microenvironment, yes, there were definitely fewer FDCs, but it looked like sharing a circulatory system with an aged animal reduced the percentage of FDCs in the adult mice as well. So suggesting, and to us, these data suggest that there's probably both FDC intrinsic, but also a hematopoietic contribution to the defective FDC response in aging. And I'm not going to talk about this today. This is something that work is ongoing in the lab to try and understand what's going on here. So what I'd like to discuss with you is these marginal reticular cells and how they've changed with age. So we did bulk RNA sequencing from the MRC population, both in the absence of immunization and 2 days after immunization, to really capture those early vaccine-induced changes. And what we could see in younger adult mice was it was clear that after immunization, the MRCs changed their transcriptional profile. So they're responding and changing in response to the vaccination. And these are some of the pathways that are enriched after immunization. So MYC targets indicative of proliferation, the interferon alpha and gamma responses, things that are well characterized to be induced upon immunization. When we looked 4 days after immunization, what you can clearly see is that in a lymph node that's responding to immunization, so the draining lymph node as compared to a non-draining lymph node in the same animal, that there are many more MadCAM positive, podoplanin positive, marginal reticular cells. And of these cells, many more are Ki67 positive, indicating that they're proliferating. They also upregulate surface expression of podoplanin and VCAM, so suggesting that they've become activated and are responding to immunization. So what about in aging? After immunization, we didn't see the same expansion in the MadCAM positive MRCs, and this was likely due to the fact that they didn't express Ki67 indicative of the cell cycle to the same extent as in younger adult mice. Likewise, they also didn't upregulate surface expression of podoplanin, so suggesting that they're not well activated after immunization. And so what we wanted to understand was, okay, so these cells don't appear to be responding after vaccination. Is there something that we could do to boost this response? And in the RNA sequencing data, what we could clearly see was that TLR4 was expressed by MRCs, as was MID88, the adaptive protein that the TLR4 signals through. This was confirmed by a flow cytometry staining that you can see here, showing that TLR4 is expressed by MRCs. We also were aware that because of their unique location in the lymph node, MRCs are at a really good spot to sort of sense immunization, just being under the subcapsular sinus. And we were also aware of this really nice work that shows that you can choose an adjuvant that can specifically target antigen to the subcapsular sinus. And so we thought that these might be, these adjuvants, particularly those that contain TLR4 ligands, would be potentially a rational strategy for boosting the MRC response in aging. We were particularly excited to, because we had been previously working on an adjuvant, or working with an adjuvant called GLA-SE, which is an oil and water immersion that, like the adjuvants that I've just shown you, can localize to the subcapsular sinus, and it contains a TLR4 ligand. We had previously run a study using this adjuvant, or comparing this adjuvant to the standard alum in people. These are younger adults, unfortunately. And we knew that when GLA-SE was used as the adjuvant, that you had a much nicer boost of these circulating Tfh cells, so really suggesting that it could target, or that it would be a good way of potentially boosting the germinal center reaction. So what Alice did was she immunized mice with either NPKLH or with NPKLH emulsified either in L-hydrogel or GLA-SE, and then had a look at the immune response 4 days later. And what you can clearly see is that in the lymph node that was given GLA-SE, you have a nice expansion of these MRCs in both adult mice and in aged mice compared to those given L-hydrogel. And the nice thing about the design of this experiment is the mice were given L-hydrogel on one flank and GLA-SE on the other flank. So you have this nice internal control where in the same animal, you can look at the different levels of proliferation in the MRCs. So you can see in both the adult and the aged mice, you get this really nice boost of the proliferation of the MRCs with GLA-SE as compared to L-hydrogel. And we wanted to understand whether this was linked to a boost in the germinal center response. So this is very, very early, day 4 after immunization. So really corresponds to this initiation of the germinal center response. And what we could clearly see was that both the percentage of B cells that were germinal center B cells and the total number were increased when the vaccine was formulated in GLA-SE as compared to alum. And so this was nice because it really suggested to us that these defects in the stromal cells with aging are rescuable, so you can actually do something about it, and that it may have an effect on the germinal center B cell response. However, when we did a kinetic of the germinal center B cell response, even though we saw that the initiation of the germinal center response was boosted by GLA-SE, we didn't see a correction at the peak. And this was also linked with not completely being able to rescue the antibody titer in aged mice. And this really led us to hypothesize that whilst these MRCs might, the MRC-related defects in aging are probably linked to impaired initiation of the germinal center, there's probably other things going on in aging that contribute to a defective peak of the germinal center response, and that's what I'd like to touch on now. We were very conscious that one of the things that has not been studied in aging was how the structure of the germinal center response changes with age. And these are the confocal images that I showed at the beginning of the talk. And you can see by eye that the germinal center looks different in aging. And what we really wanted to do was quantify that. The germinal center is divided into 2 functionally distinct zones, the light zone and the dark zone. In the dark zone, germinal center B cells proliferate and undergo somatic hypermutation of the genes encoding the B cell receptors. They then migrate to the light zone, where they interact with follicular dendritic cells and T follicular helper cells to receive positive selection signals that then allow them to migrate back to the dark zone using CXCR4 to either undergo the cycle again or exit the germinal center as antibody-secreting plasma cells and memory B cells. So we did quantitative imaging of how the germinal center changes with age. And what we saw, one of the things that we saw that was really interesting was that when you looked at the light and the dark zone and then counted the T cells in either of the zones, what you see in a younger adult animal is that the T cells, follicular helper T cells, are localized in the light zone, which is what any review on follicular helper T cells will tell you. And that there are some T cells in the dark zone. So you can see that there are some, but they're mostly enriched in the light zone. Now, interestingly, we didn't see this enrichment in aged mice, and you can see that they look much more dispersed through the germinal center. And what Alyssa and Sigrid did was quantify this. So each dot here is a biological replicate where they've measured multiple germinal centers and then are showing the average in this dot. And what you can clearly see is that in 22-month-old aged mice, there are more T cells in the dark zone area of the germinal center. And we hypothesize that this means that the TFH cells are lost and that the help isn't where B cells need it. So they need help from TFH cells in the light zone where they've met the FDCs and are presenting antigen, not in the dark zone when they're undergoing rounds of proliferation and mutation. It does also highlight that under normal circumstances, you do see TFH cells in the dark zone, which is something that is underappreciated. So we wanted to understand why the TFH cells might be mislocalized in aging, and what's known about the movement of germinal center B cells in secondary lymphoid tissues is that they up and down regulate the expression of the receptor CXCR4, whose ligand CXCL12 is expressed in the dark zone. So when they're high for CXCR4, they are dark zone localized. When they downregulate CXCR4, they can migrate to the light zone, responding to CXCL13, a chemokine that binds CXCR5, which is expressed both by TFH cells, but also by germinal center B cells. What was very clear in aging is that aged mice had much higher frequency and number of CXCR4 positive TFH cells. And if you take these TFH cells out and you do a chemotaxis assay, what this equates to is that TFH cells from aged mice are more likely to migrate towards CXCL12. So indicating that this increased expression of CXCR4 actually translates into an increased functionality of that receptor. And so these findings prompted three questions for us. One is that how is TFH cell polarization controlled? So it suggests that CXCR4 might actually control the localization of TFH cells within the germinal center. It prompted the question of how does TFH localization influence GC or germinal center responses after vaccination? And also led us to hypothesize that if you could correct this mislocalization of TFH cells in aging, that you might be able to correct the defective peak of the germinal center response. And so this work was led by Alyssa Silva-Cayetano, and to answer the first question that we had, does CXCR4 is CXCR4 functionally responsible for the localization of T cells? We used OT2 T cells in which CXCR4 could be deleted upon tamoxifen administration with a tamoxifen-inducible CRE. So in vitro, she treated the cells with tamoxifen for 48 hours. Interestingly, for CXCR4, at this point, this is enough that the cells are mRNA negative for CXCR4, but still have some surface expression, so they don't lose the ability to migrate until they've undergone a few rounds of division. We transferred those into congenically distinct mice and then had a look 10 days later. And what was really clear was that in the control animals that were just CXCR4 floxed, you could see T cells both in the light zone and in the dark zone, as marked by CD45.2 in magenta. The light zone is marked by the follicular dendritic cell network of CD35 in white. And what was really striking in these experiments is we could rarely see cells that had lost CXCR4 in the dark zone, and that's just enumerated here. So when CXCR4 is deleted off T cells, they're mostly localized in the dark zone, in the light zone, with a corresponding decrease in the dark zone. So then this really begs the question of, does this change the biology of the germinal center response? So we made another strain of mice, so CD4 CRE, CXCR4 flox. So in these animals, all T cells will lack CXCR4. And again, what we could see was that the majority of T cells were in the light zone with a corresponding decrease in the dark zone. But interestingly, this didn't change the area of the germinal center, so how big the germinal centers were. And so from these experiments, we can conclude two things: that CXCR4 actively controls TFH cell position within the germinal center, but we wanted to then ask the question, are there any implications for the output of the germinal center response? And what we found was quite surprising. So even though there was no difference in germinal center size or indeed germinal center B cell number, what we found was that in these animals, there was an accumulation of high affinity germinal center B cells. So in the NP system, this is denoted by the acquisition of an L at position 33 compared to the wild type W. And this was reflected in antibody levels. So the total serum antibody levels were comparable between mice whose T cells lacked CXCR4 or were sufficient for CXCR4. But interestingly, there was more high affinity antibody in the sera, and this corresponds to an increase in affinity maturation. So what about if you do the opposite experiment? You know, what we really wanted to do was see if we could phenocopy the aging phenotype by skewing T cells away from the light zone. And for this, we took inspiration from a paper from Jason Sister's lab, who had previously shown, using adoptive transfer of CXCR5 deficient T cells, that CXCR5 was really essential for the light zone polarization of follicular helper T cells. So in the control animals, the majority of T cells are in the light zone, whereas in CXCR5 deficient, mice that received CXCR5 deficient T cells, they were dispersed throughout the germinal center, so very similar to the aging phenotype. So what we did was we made some mice where we used the CXCR5 floxed allele and crossed to the CD4 Cree allele. So again, all T cells lack CXCR5. And what was quite neat was that in this system, which is a different biological system to that of the sister lab, we could replicate their finding that there were fewer T cells in the light zone of the germinal center, with a corresponding increase in T cells in the dark zone. So then what does that do to the germinal center? And what was really nice was we could clearly see that the number of germinal center B cells was diminished when CXCR5 was lacking from T cells. And this also corresponded to a reduction in antibody production. So very similar to what we saw in the aged mice, really suggesting that TFH cell positioning is important for the germinal center response. So that really led us to hypothesize that the supply of T cells that could correctly localize to the germinal center light zone could correct the aging germinal center response. And to do this, what Alyssa did was she took ovalbumin-specific OT2 T cells and transferred them from a younger adult animal into either younger adult or aged mice immunized with NP-OVA, and then had a look 10 days later. What was really nice is that by confocal, what you could clearly see is there are nice germinal centers in the aged mice that received OT2 T cells, and you can see that they've localized. The transferred cells, which are marked in magenta, are mostly localized to the white FDC network. And this is just enumerated here that younger, or T cells from a younger animal have correct localization in an aged host, which is good because it allows us to move to the next step of the experiment to see what the implication for the germinal center response is. But it also tells you that whatever is causing the defective localization of TFH cells in aging is not imprinted by the aged microenvironment. So it's probably something that's intrinsic to the T cells. So in this experiment, by transferring these OT2 T cells, what we could clearly see was that the area of the germinal center was increased. And if we looked by flow cytometry, what we could see really nicely was that there was an increase of germinal center B cells, both as a percentage and total number at the peak of the response. Interestingly, at day 7, which is much more about the initiation, we didn't really see a strong effect of the OT2 T cells being able to correct this defective initiation of the response. So again, sort of supporting this idea that there's probably one defect that underpins impaired germinal center initiation with age, and another defect, age-associated defect, that underpins the reduced size in aging. So we were very excited by these results, But what we really needed to do was a important control experiment to show that it's not just transferring young T cells into an aged mice that has the effect, that it's really about transferring T cells that correctly localize to the light zone. And so what Alyssa did was she transferred either wild type OT2 cells from a young animal into an aged mouse, or OT2 T cells that lack CXCR5. So that both Jason's sister and then we subsequently confirmed can get to the germinal center. So they can be in the germinal center, but they don't localize to the light zone. So really allowing us to ask this question about, do these young T cells need to be light zone localized? So here's just showing you that the CXCR5 staining is present on the control T cells, but not on the knockouts. And then what you can clearly see is that when aged mice are supplemented with CXCR5 knockout OT2 T cells, you don't have the same correction of the germinal center response 10 days after immunization. So really supporting the idea that T cell localization or defective T cell localization in aging impairs the germinal center response. So just to summarize this part, what we've shown is that CXCR4 actively controls germinal center localization and localizes a proportion of TFH cells to the dark zone, which in aging, because of increased CXCR4 expression, means that you have more TFH cells in the dark zone. Interestingly, the loss of these dark zone TFH cells by removing CXCR4 actually results in enhanced germinal center affinity. So having more T cells in the light zone improved the affinity of the response, which was a surprising finding to us. We were expecting the opposite result. And importantly, we were able to show that supply of T cells to the light zone corrects germinal center defects in aging. So really suggesting that not only does localization matter, but it particularly matters in aging. And so finally, I'd just like to acknowledge everyone who's contributed to the work. As I said at the beginning of my talk, nothing happens without an amazing team. In particular, the work presented today was really spearheaded by Alice Denton, the first part of the talk, Alyssa Silva-Cayetano, the second part of the talk, and Danika Hill, who did all of the circulating TFH work that was shown in the talk. So thanks for tuning in. Pleasure to be here.
Thank you very much, Michelle, for a wonderful talk. And let me remind everyone that the question will be asked via Twitter, as always. And don't forget to connect for next week's seminar about effect Ordnung. Thanks again, Michelle. And bye, everyone.